Welcome back, friends. In today's session we are going to continue with our coordination topic, and today we are going to discuss we are going to continue our discussion in Auxins. And for the case of today we are going to see how different function of Auxin, which we have discussed in the previous session, we are going to see how can they be seen diagrammatically, how are they affecting the plant. Now let's see, in our first diagram we have the effect of auxin in phototropism and geotropism. Now here we have the shoot of the coleoptile. So coleoptile ni aina mmea, sio kujua what is coleoptile. Coleoptile ni aina mmea, na ukija kuangalia shoot ya coleoptile hapa ina different cells. Unakuta kuna cells huku left side na huku right side kuna cell, na pia hapo kuna cells. Kwa unachokiona ni kwamba in the absence of light, easy auxin they are produced at the coleoptile tip and they are distributed downward, kama tulivyosema kule mwanzoni. Kwa auxin they are produced here and they are distributed downward. Now as we said kwamba the auxin they tend to stimulate cell elongation. So following their distribution to downwards the plant, they stimulate cell elongation, and as you can see the coleoptile become longer. So we have auxin move to the shaded side within the tip, they move downward, and then redistributed auxin move down the coleoptile. So they are moving down the coleoptile. Here we don't have light. So in the presence of light, now in the case of phototropism, in the presence of light, if there is no light, the coleoptile tend to elongate, but there is no bending. In the presence of light, now as I told you that the distribution of auxin, it tend to escape from sunlight, so it is distributed on the shaded side. Kwa hiyo auxin from the tip will be distributed more to the shaded side. And as you can see in this diagrammatic presentation, the cells on the right hand side, they have elongated, they are shorter as compared to the cells on the right hand side. So the cells, on the left hand side, the cells on the left hand side, they have elongated more as compared to the cells on the right hand side. So because the cells on the left hand side, they have elongated more, and remember the direction of our illumination is from this direction. Now because the cells of this shaded side, they have been elongated more, that means auxin, they have a direct effect on phototropism. So if we don't have auxin, we don't have phototropism. Without auxin, no phototropism. Without auxin, phototropism cannot occur. So as you can see, a high concentration of auxin causes more rapid growth by cell expansion on the shaded side. The tip curves towards the light. Now it is because there is a light on this side, and then illumination of this side by light, it causes the bending of the tip, and the bending, it is because of the cells on the shaded side expand more than those on the other side. Why? It is because these cells on the shaded side, they receive a higher amount of auxin as compared to the cells on the light side. Now again, if the coleoptile tip, it is horizontally directed, manake in this case coleoptile tip zilikuwa, the coleoptile was vertical, and we see the effect of auxin on phototropism. But if the coleoptile tip now it is in a horizontal direction, assuming here the seed is germinating, and when the seed is germinating, the coleoptile tip, it is in a horizontal direction. Now what causes the shoot to move upward and the root to move downward is because of the effect of auxins. How? It is because, as you can see in this diagram, here we have the cells on the shaded side and here on the upper side, and because the sun illuminates from above here, now the cells on the lower side, they will be on the shaded side, and then the cells on the upper side will be illuminated. Kwa tukipata jua huku juu, kama tulisema kwamba distribution of auxin inakimbia mwanga, so more auxin which are produced on the collected tip will be distributed on the shaded side. So auxin move downward in response to gravity, and not only gravity, it is also because here most of the time the seed, it is buried downward, so it is not easier for the sun to reach the collected tip. But when auxin, they are moving downward, and then moving auxin to downward will lead to the cells on the downward side to have higher concentration of auxin as compared to the cells on the upper side. So this cell will elongate more, and the elongation of this downward cell more will cause the bending of this collected tip towards the side where the cells, they have not elongated more. So a higher auxin concentration causes more rapid growth on the lower side. The tip curves upward. So as you can see, in the phototropism, distribution of auxin is affected more by light, and it tends to escape the direction of light illumination. And in the geotropism, distribution of auxin, it is because of gravity. Gravity tends to pull all the things towards the center of the earth, and here it tends to attract auxin downward to these cells. Now let's see again this diagram, the effect of auxin, I mean the effect of light on auxin distribution. So as you can see, if we have the sun here, and then it illuminates this collected tip, when sunlight is overhead, the indole acetic acid, that is the most common type of auxin molecules produced by the apical meristem, are distributed evenly in the shoot. So they are distributed evenly, that means kwa usawa. So the distribution on the left and right hand side, it is equal. But once the sunlight shines on the shoot at an angle, manake kwenye upande mmoja au illuminated at an angle, the indole acetic acid molecules move towards the far side. The far side manake upande ule ambao kunakuwa hakuna jua, so moves towards the far side and induce the elongation of cells on the side, on that side. Kwa hiyo at the end of the day unachokuja ukiona ni kwamba huku kuna low concentration of auxin, kwa sababu auxin zinakimbia mwanga, zinakuja kuwa distributed on this side. So distribution of auxin on this side will lead to the more cell elongation on this side, and at the end of the day we'll end up with the bending of the coleoptile tip. So cell elongation results in bending of the shoot towards the light, and this graph tends to demonstrate the effect of auxin in roots and shoot. This is how the graphical representation can be seen. So as you can see here, we have zero concentration. We have zero, I don't mean concentration, I mean it is a zero growth percentage. So from here to upward it is a stimulation, from here to downward it is inhibition. So in zero concentration, these are just concentration of auxin. In 10 power negative 5 concentration of auxin, it doesn't stimulate anything. And then in 10 power negative I mean this is 10 power negative 6, in 10 power negative 5 it starts to stimulate the root development, and in 10 power negative 4 it stimulates root development and elongation. But as you can see here, this is the concentration which affect the root development, tend to stimulate the development of root in very low concentration. However, at this concentration it does not affect, or it does not stimulate at all the development of shoot. So the graph of development of shoot here is zero. That means it only stimulates the development of roots. And at this point, as you can see, this is the level of auxin secreted from roots. Level of auxin, this is the level of auxin which start to stimulate the shoot development. And as you can see, the maximum level of root development stimulation doesn't stimulate the shoot at all. But as you can see also that the level of auxin which start stimulate the shoot development, it start to inhibit, because this graph of root development start to fall and the graph of shoot development raises. That means the concentration of auxin in this horizontal line in x scale, if the concentration of auxin increases, then the root development is inhibited. For example, this auxin concentration, root development is inhibited at all. But as you can see, at this concentration, it is where the shoot development is stimulated more. So the level of auxin stimulated by roots, it is almost this very low concentration. That's why we told you before that it is excreted from the tips of it is excreted from the shoot apex and young leaves. However, very small concentration of auxin can be produced from roots. As you can see, this concentration is very small and it is insignificant on the growth of shoots, but the level of auxin secreted by stem or by the shoot apex, it is very high, which tend to inhibit the growth of roots. So the amount of auxin required by the plant in order to stimulate either shoot or root development, it is different from these two cases. Now let's finish our discussion by discussing this diagram. In this diagram, this is the effect of auxin on roots. And then here we have two orientation of roots. We have the vertical orientation and we have the horizontal orientation of roots. Now in the vertical orientation of roots, the distribution of indole acetic acid is synthesized in the shoot and transported to the root in the stem. So it travels through the xylem tissue to the root, and then after reaching the root, the stage number 2, when the root is vertical, the staphylococcus in the cup settle to the basal end of the cell. Auxin transported acropetal in the root via the stem is distributed equally on all sides of the roots cap. The indole acetic acid is then transported basipetally within the cortex to the elongation sides where it regulates cell elongation. Unaweza kuona ni kwamba hii auxin kwanza ilikuwa transported, tuisema the movement of auxin is polar, that is one direction. So unidirection mpaka ikafika hapa. This is staphylococcus, and the staphylococcus they act as molecules ambazo zikireceive auxin at the root cap, they tend to start distribution of auxin again to the region of cell elongation, because as we said that the movement of auxin, it is in one direction. Kwa direction hii hapa ikianza hapa lazima ifike mpaka kwenye tip, and as we said the movement is also basipetal, that means it comes from the tip to the other region of cell elongation. Kwa unakuta hii ni root cap, na tukiwa na root cap, auxin zikitoka huko lazima zifike kwanza hapa kwenye staphylococcus. Zikifika kwenye staphylococcus zinaanza kuwa distributed basipetally from the staphylococcus or from the root cap back into the region of cell elongation. Now here we have the zone of elongation, where we have the flavonoid synthesis, and then here we have the cortex and the style here, which tend to enable the movement of auxin from the shoot. So here, because the auxin they have distributed equally on both sides, the root development will develop just vertically downward. because the stimulation of the cells on the right and left hand side, it is equal, they are equally stimulated, so the root will develop downward. But in this diagram, the roots, they are horizontal position. So in horizontal position, the auxin traveling from upward, when they are going down to this stathoris, you see we will have an increased distribution of auxin below this one. So the majority of auxin in the cape is then transported basipetally in the cortex on the lower side of the root. And as we said, this is probably related with the gravity, because auxin, they are molecules of indole acetic acid, and even if they have lower masses, but they can be attracted by gravity. So gravity pulls more auxin to this lower side. And in horizontal root C, the stathoris settle to the side of the cape cells, triggering polar transport of indole acetic acid to the lower side of the cape. So because this is attracted also by gravity to the lower side, and most of auxin is distributed in the lower side, so the higher concentration of auxin on the lower side will inhibit growth, will inhibit root growth. As we said, we discussed in the previous graph that the higher concentration of auxin tend to inhibit root growth. So by that way, that means on this side the root will grow less as compared to this side. This side the root will grow more because we have low auxin concentration, which tend to stimulate root growth. So the decreased auxin concentration on the upper side stimulates the upper side to grow. As the result, the root bend downward. So this is the effect of auxin in the vertical and horizontal roots orientation. So normally if the seed germinates, it is also the shoot lies horizontal and the root lies horizontal. Now if the root lies horizontal, that means distribution of auxin will be more on the left, on the lower side, and then the roots will bend downward, and again the roots will be regarded as positively geotropic. But why are they positively geotropic? It is because of the effect of auxin. But the shoots, the distribution of auxin to downward because of gravity will stimulate shoot development and bending the shoot to upward. That means it bends to up, and it is described as negatively geotropic or positively phototropic. But all in all, this is the effect of auxin. The session was somehow very long, but I hope we have understood each other. If you have any question, you can comment this video, or you can join my Telegram group for any question, or even you can see my contacts and then you can contact me through either WhatsApp or Telegram. Thank you everybody. Don't forget to subscribe the channel for more updates, and also don't forget to share this video and to like this video. Thank you. Let me wish you nice studies.